Welcome to the DiabetesBio podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion, additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk and Dr. Kevin Williams. How are you guys doing today?
Hey, Dar.
Hey, doing good.
Can you believe we've been doing this for a whole year now? Congratulations.
Woohoo, yeah, it's amazing.
They have not kicked us off the air. It's very impressive. So to celebrate our 1-year podcast anniversary, we've selected some titles of articles in the journal that we loved, but just didn't have time to interview the authors. So Kirk, let's start with you.
Yeah, I guess I wanted to start with establishment of pancreatic beta cell specific gene knockout system based on CRISPR-Cas9 technology with AAV8 mediated gRNA delivery. That's a mouthful, but it's really cool work from Chantendo University Graduate School of Medicine describing the use of CRE to express Cas9 in a conditional manner, following that with AAV delivered guide RNA against your gene of interest.
These tools are getting fancier and fancier.
Yeah. And, maybe we're not beholden to whether we can get the Flox mouse again.
Yeah.
Right. The second article I wanted to mention was the effect of small increases in blood glucose on insulin secretion and endogenous glucose production in humans. This is work by Bruce and colleagues at Deakin University in Australia. Here, the authors conclude that small glycemic increments exert subtle stimulatory effects on insulin secretion that may have potent metabolic actions on the liver and adipose tissue.
Awesome. Kevin, how about you?
Yeah, those are great suggestions, Kirk. It has been a great year. I first want to give a quick reminder about the Diabetes Symposium that was held at this year's ADA annual meeting in June. We had 4 great speakers, Christy Townsend, Zaman Mirzadeh, Laura Ruprecht, and myself. And for the first time, all these presentations were recorded and are available in this month's issue. So be sure to visit the Diabetes Journal website to read the articles and watch the presentations. For a couple articles we did not get to earlier this year, I'd like to start with GIP receptor agonism enhances TZD-induced insulin sensitivity in obese insulin-resistant mice by Ricardo Sams and colleagues, and it's in the February issue. This article explored how GIP receptor agonism improves insulin sensitivity while preventing weight gain associated with TZD treatments.
I'm really glad you mentioned that one. We tried to get Ricardo to join us on air, and it just didn't work out timing-wise, so that's a cool article.
Yeah, I totally agree. And yeah, I was unfortunate with the timing issues that we had there. So yeah, it's good to highlight it here. And in the November issue, we had evolving concepts in pathophysiology, screening and prevention of type 1 diabetes, a report of Diabetes Interagency Coordinating Committee workshop by Carla Greenbaum and colleagues. They highlighted the approval of a therapy that delays type 1 diabetes onset, teplizumab, and discusses strategies to improve its clinical application.
Awesome.
I think both of these articles are topical and a great read. Dar, what about you?
So I liked the role of cardiorespiratory fitness and mitochondrial oxidative capacity in reduced walk speed of older adults with diabetes by Ramos et al. In the July issue. And these are folks at the Translational Research Institute in Orlando. And so I don't know about you all, but as I get older, how to maintain my mitochondrial oxidative phosphorylation is pretty important.
Absolutely.
And then another one that I liked was, and this I liked more because of that's sort of the experimental technical strategies, was development of novel tools for dissection of central versus peripheral dopamine D2-like receptor signaling in dysglycemia. And this was by Bonifazi et al. I apologize if I mispronounced that. And it was in the June issue. And I just love their ability to be able to tease apart the central and peripheral actions of this drug.
Yeah, very cool stuff.
Kirk, why don't you tell us what we have in stock for our feature paper interviews?
Yeah, so this month we're going to discuss the paper Microglia Mediate Metabolic Dysfunction from Common Air Pollutants through the NF-kappa B signaling. That discussion is with lead author Lukas Stilgenbauer and senior author Mariana Sadagurski. We then chat with lead author Max Petersen of the paper Effect of Weight Loss on Skeletal Muscle Bioactive Lipids in People with Obesity and Type 2 Diabetes.
Kevin, what's happening for our Sweet Talk segment?
Yeah, today we're joined by Dr. Kathleen Page, an associate professor of medicine at USC's Keck School of Medicine, and Dr. Jasmin Alves, a postdoctoral fellow that works closely with Dr. Page. They're discussing the recent article in this issue of Diabetes, which highlights how early life experiences shape our metabolism, focusing on the role of neurodevelopmental pathways in the development of obesity and diabetes. This work was also supported by the American Diabetes Association Pathway to Stop Diabetes Initiative, an award that Dr. Page received in 2014. So stay tuned for this interview coming up later in the podcast.
Awesome. Let's jump in. Today, we're going to discuss a featured article titled Microglia Mediate Metabolic Dysfunction from Common Air Pollutants through NF-kappa B signaling. We are joined by lead co-author Lukas Stilgenbauer and senior author Dr. Mariana Sadagurski. Dr. Stilgenbauer recently defended his thesis. Congratulations. And Dr. Sadagurski is Associate Professor in the Institute of Environmental Health Sciences and Integrative Biosciences Center at Wayne State University. Welcome, Dr. Sadagurski and Stilgenbauer. Congratulations on your paper, and thanks for being a part of this month's podcast.
Yeah, thank you for having us.
Thank you very much for inviting us. We're very happy to be here today.
All right, so if you've listened to the podcast, we start with an unscripted icebreaker question, just so the listeners can get to know you. So I know, Luke, that you just defended your thesis, but if you were not going to pursue a career in science, what do you think you would have pursued?
It would probably be something similar to science. So I'm a big wild mushroom hunter. So out here in Michigan, we have a good amount of wild mushrooms, and I like to hunt them. I've sold them to restaurants a couple times before. And it's fall right now, so we have a good like hen of the woods and chicken of the woods kind of season going on right now.
Awesome. And Mariana?
Oh my goodness. I think it's been so many years already in science, it's very difficult to say anything else that is not science related. I almost speechless. Well, I started running like on a weekly base. We have in Ann Arbor what's called park marathons of 5 to 10 Ks, and I started doing it on a weekly basis, and I'm very excited about my progress.
All
So
right,
that's like a new
awesome.
thing for me, which is not related to science, but very
That counts.
excited.
That's awesome.
All right, let's get to the paper. Mariana, first we like to give our authors an opportunity to highlight any other contributing authors or collaborators to this work. So is there anyone you want to talk about right now?
Yes, so this work started, I would say 4 years ago, maybe even 5, when a postdoctoral fellow, Lukas Stilgenbauer, joined my lab. He came from Brazil, University of São Paulo. And we were very interested to start pursuing this sort of new direction for me, which is environmental exposures. So Lukas was brave enough to start this project from zero, to pick it up, to bring it to where it is right now. And, we have so many different other avenues in my lab, sort of, coming from that direction. And it's all thanks to him and to what he started in our lab. And he's actually the first author, but he already left the lab. And now he's in New York in NYU. So he's not actually joining us, but I think that lots of thanks actually goes to Lukas, because he helped us to start this new adventure.
That's a great introduction to this. You said this was an offshoot of your normal research. Can you maybe give us an overview of your research program and then how this specific project fits into that?
Yes, so I'm a trained physiologist, and we're mostly looking at the central nervous system and how hypothalamus specifically regulates peripheral metabolism, glucose homeostasis, and energy expenditure. Now, usually you look at the way hypothalamus responds to and how hypothalamus regulates periphery in responses, for example, to obesogenic factors, diet-induced obesity, and so on. And what we took as a sort of direction from that in my lab is to look at how different environmental factors, environmental pollutants in the air, can predispose hypothalamus to some sort of stressor, sometimes continuous stressor, sometimes more acute stressor, that then predispose us to develop more sort of metabolically vulnerable state of our body. So we're looking at environmental factors, and we're also interested how different environmental toxins, if you expose through your lifespan, how they also affect the aging process. And going back to the hypothalamus, as we age, we're also more prone to develop hyperglycemia, insulin resistance. We are more metabolically, weaker, right? And lots of this has to do with our nutrition, with lack of exercise and so on. But we are actually more sort of focusing on environmental factors and how these sort of make hypothalamus more vulnerable to these long-term stressors, and as a result also predispose us to more metabolic vulnerable state of our body. So this is what we're doing. And I think we have been very lucky here, because here in Detroit, we have these environmental centers that allow us to be, more bold with these questions that us as metabolic researchers were not used to ask. And we're not used to even look at that from the perspective of toxicologists, because we're not.
Sure.
So this particular location of ours in Detroit, which is where we are, and the centers that we have here, which is one of them, it's Cures, it's Environmental Health Sciences Center, that actually has all these capabilities that allow us to start asking questions and actually bring, diabetes research to environmental field world, as well as bring environmental field world to diabetes research, so sort of mix and match.
Yeah, it's a very cool crosstalk.
One of the things I liked about your study is that you start with a meta-analysis of the literature. And I thought that was a really creative way to bring that translational aspect to the project without having to actually have clinical research partners and do a very expensive clinical research study. But you found a robust association in the literature between benzene exposure and insulin resistance. And that seemed to occur across all ages. Do you have any, why do you touched on this a little bit in the previous question, but why do you think benzene in particular is important, and what do you think the implications are for this finding?
So I will start from the beginning, and that is, again, for us that are not a toxicologist, we sort of assume that every single toxin that we have should probably trigger metabolic disease or have some sort of effect on metabolism. We don't really know, but we assume, right? Because for us, it's sort of obvious. So here specifically, and it's not just in Detroit, in basically every urban environment, you have very high levels of VOCs. VOCs are volatile organic compounds that, they're sort of like silent killers, because you have them everywhere. You have them in waters, you have them in your food, you have them, gas stations and so on, that's obvious. But here we have another very serious problem, and that's what's called a vapor intrusion, which means that people's basements many times, and the houses that people have, they're built on top of whatever was left behind from the car industry, for example, from 20s, 30s, and so on. The levels of these VOCs in people's basements, these vapor intrusions, many times significantly higher, 10 times or more higher than what you actually have in your air in the environment, even, again, going back to the gas stations. So us thinking that, if our air outside is clean and that's enough, that's good. However, these vapor intrusions, they're still there, right? And people just live there. And the levels in the basements are very high. Well, again, in Detroit, for example, you have certain zip codes where it's many times higher rather than in other places, which is, of course, a critical problem to understand. And, we have high rates of type 2 diabetes here, not necessarily directly going with obesity. And it's not genetics, right? So is it all just nutrition, or is it these other factors that you have here at a very high? And that is why for us, VOCs are so important. Specifically, you asked benzene, going back to that question. Well, we needed to start with something, right? VOCs are mixtures. You have many of them. Again, sort of going back to Detroit as one of the serious problems that we have here, we have these combinations that are called BTEX, benzene, xylene, toluene, isobenzene, that are found to be very, very high in these areas. And again, when we say high, the concentrations are crazy low, and they are approved levels to some extent, but they're much higher than in other areas. And you need to start from somewhere, right? It's very difficult to say, okay, all of them are bad. How much enough is enough? When are these levels of, when you reach this difference that you're already hyperglycemic, you have changes in your metabolic homeostasis. So we needed to start with something. Benzene for us was a good example also because of its association with smoking, how much you have in the cigarette smoke. So we went from there, starting sort of higher, and now we're going lower because you needed that threshold. And when I said that for us as metabolic researchers that assume that everything bad triggers metabolism, that's why it's obvious, we needed that meta-analysis, because when I came here to toxicologists saying, well, let's look at diabetes because it's obvious, they said, well, do you have actually epidemiological data to support that? And I figured, well, for sure, why not? It was for me, I didn't even think, also because I was not used to look at this type of data. And we actually started searching, and we realized it's not that trivial. There is not that much data out there. Most of that was PM 2.5, which are certain particles. There's lots of literature out there. But these VOCs, well, they evaporate, right? These are volatile, they evaporate. So it's very difficult to detect. And that's why there's not much data. And for us, it was critical to look at for that data, to start searching who said what, how many studies are there. And then we discovered that there are studies that are showing correlation between HOMA-IR, which is index of insulin resistance, and, for example, metabolites. So you can detect these volatile organic compounds, metabolites, in people's urine, right? This is what our meta-analysis is doing, basically. So we collected all the data from papers that they had. And actually, Luke was the one who did all this screenshot of every single paper. And then we did all this, who actually measured in urine, in the blood, what metabolites of these VOCs they had. And then whether they had measures for the HOMA-IR, or, for instance, just fasted insulin, fasted glucose that we can plot ourselves. So there was a lot of work for that, to get that done, because there is not that much. And once we got that, and I think that our total number of people that from all these studies, it's about 25,000 people total of different ages, from young adults, just adults, and even aged populations. There were some studies in Korea that we found. And all that emphasizes that it's actually a critical problem, not just on toxic levels, but actually environmental levels, which is what you just have here in Detroit as well. I'm sorry, it was a long answer, but we feel very passionate about that.
And just kind of touching again on the idea of why benzene. So Mariana brought up that it's an environmental toxicant. It's we call it a ubiquitous environmental pollutant because it does seem to be everywhere. So, you have gasoline, you have combustion, you have cigarette smoke, but you also have these kind of newer problems that we're dealing with. Specifically, it's also found in e-cigarette vapor. So this can be a link towards possible metabolic dysfunction from that. And you have it off-gassing from polymers, cleaning products. So it's kind of insidious because benzene, even in low amounts, is in a lot of places you might not even expect it to be. So it's a good stand-in for just VOC exposure in general.
And that is a great way to bridge into our next question, right? So you've performed studies in mice to determine the impact of benzene exposure on specific aspects of metabolic homeostasis. Now, previously you saw no impact of benzene on body weight, right? But you did see several indicators of alterations in metabolism. Can you maybe tell us first how you performed the benzene exposure, and then what were those critical impairments on metabolism?
Right. So what we have, these environmental chambers in which animals just sit in a regular chamber as they usually are sitting. They're housed in the same way. Everything is the same. It's just whole body inhalation. So they can inhale these toxins, these VOCs, at the concentrations that we define. It's a computerized system, basically, that's built by company, permeation tubing, which means that we can control how much gets in and the dose, the concentration, and the time course. Now, these VOCs are already been defined previously through these environmental studies as being endocrine disruptors. So these, and benzene is one of them, these are endocrine disrupting particles. And as a result, you would anticipate to see changes in body weight, right? Now we continuously, even over the period of up to 6 weeks, which was our previous study, we didn't see changes in body weight at all, which was a huge surprise for us, even disappointment at the beginning. But that also might indicate that our concentrations are very low, and it's just not enough to pick up the body weight changes, which is cool, because we also were not able to see any changes in blood levels, like, red, white counts. So there is no cancer-related changes either, which is these toxins are known for. So we're not triggering any carcinogenesis at all at any age or time frame that we're doing our studies at least, and no changes in body weight. Now, metabolically, the biggest change was blood glucose levels. We see almost after 4 days of exposure, we start seeing these peaks of blood glucose. We're doing what's called, we're using CGM, continuous glucose monitors, in which we're just implanting the sensor of the CGM underneath the skin of these animals. And this we're doing with my collaborator, who is also a co-author on this paper, Ulrike Klu. She's a biomedical engineer who is working on these devices. So once we started sort of looking at the blood glucose levels, we were able to see that already at the 4th day of exposure, there are changes in blood glucose levels of these animals, and these changes just keep going and going. These animals also exhibit later sort of hyperglycemia once you measure them with glucose tolerance tests. They have high levels of insulin in their blood, significantly higher than their control litter mates, and these animals also have impaired energy homeostasis parameters, such as oxygen consumption, carbon dioxide production, and energy expenditure measures in general. And these are visible almost immediately after the first exposure. So once we're exposing during the light cycle and measuring energy expenditure during the dark cycle, we can immediately detect that there is a significant drop in that. And initially you can say, well, it's maybe also stress related, right? Because that's also what we thought. So Lukas continuously also measured the cortisol levels in the blood of these animals, trying to see whether there is a correlation, and if they're continuously, the stress levels are high, and that's why it affects energy expenditure. And that's not true. So yes, it peaks at the beginning because, animals are stressed, they're not happy. But it's just inhaled and they get used to that. And once cortisol levels are normalized, the changes and impairments in hyperglycemia, hyperinsulinemia, and impaired energy expenditure just keeps going as the exposure goes. So this is our main sort of metabolic changes, while again, body weight, not even once we saw any changes whatsoever, which is exciting because it's a different sort of mechanism as well.
Right, absolutely.
Luke, why don't you tell us about you then went to look for some mechanisms, and you went to the brain, and so you found some impact on the hypothalamus and specifically an impact on the microglia. So why don't you tell us a little bit about those results?
Yeah, so we found that the microglia were inflamed following a treatment with benzene, and then because of that, we then looked at specifically, could we do something to those microglia to kind of rescue this model from this metabolic dysfunction that was seemed to be caused by these microglia in the brain.
Great.
Follow up, Mariana?
Yes. So hypothalamus is the hypothalamus, and that was also driven by our previous study. It seems to be crazy vulnerable to the exposures, right? It is very sensitive to diet-induced obesity. It's also very sensitive to, even if you put animals on high-fat diet on a short period of time, they already develop microgliosis, which is what Luke was referring to, activation of the microglia. But what we wanted to understand is whether, similar effects on pollution. And once you inhale anything, right, it goes through olfactory bulb, it gets to your hypothalamus. Once you inhale, it also affects your lung. Any inflammatory or whatever, right, it gets to your lung. Once you inhale environmental toxins, pollutants, whatever, it affects your lung. Your lung starts secreting inflammatory cytokines. For them to get to your hypothalamus is not a big deal because you actually don't have a very well-protective blood-brain barrier in that area, right? Making this especially vulnerable to anything that comes across. And that is what Luke was referring to, because when we looked at the microglia, which are brain immune cells, we don't have macrophages in the brain, but we have microglia, right? Which is our immune cells. They're also memory cells. So when Luke looked, sounds fun. When Lukas, we have Lukas and Luke in the lab, so we had both. So when they looked at the microglia in the hypothalamus, they saw that these cells become very quickly activated, inflamed, which is what you would anticipate from the immune cells, and that comes very rapidly upon exposure. Now, in his previous study, Lukas already asked whether benzene metabolites within these microglia actually activated, sort of specifically responding, and the answer was yes. So, for instance, microglia also exhibit activation of cytochrome P450, which is one of the main activators and metabolites from VOCs, not just benzene, but from others as well. And microglia do express cytochrome P450, and it gets activated, and that he showed in his previous study. So that is why even here we actually looked at the microglia, asking whether similar sort of activatory trigger stressor, as previous studies showed with high-fat diet, is also shown with the microglia upon these exposures. And the answer was yes. So Luke and Lukas looked at these microglia, and they got, they started changing shape, their morphology was changing, they were secreting inflammatory cytokines, and that was pretty quick. It was at the same time when we saw changes in glucose and energy expenditure. So this is why for us, hypothalamus was that's important, because, there were so many studies, as I'm sure in your podcast, maybe you touched base previously, right? As high-fat diet can activate hypothalamic inflammation that happens quickly. And that can be one of the drivers for metabolic impairment. So that was our question. Is it to some extent similar, right? Whether stressors are stressors, and what happens to the microglia is just so critical, and so like you cannot change that, right? Once microglia are activated, they just keep going, and inflammation there just keeps pouring. And as a result, it affects peripheral metabolism, and animals, basically sort of in a state of prediabetes, right? So that was one of our rationales of going to the microglia, more specifically, I would say. And yes, Lukas and Luke. So they looked at that after we saw these changes in the morphology and the expression of the cytokines. This is when Luke went to look at the transcriptional profiling of these microglia within the hypothalamus, actually asking what changes in these cells and how quickly these changes are, right? Because if you see something on the chronic effect, you actually don't know, right, who is the egg, who is the chicken, how quickly things are happening. But if you actually do it pretty quickly with what he did with the transcriptional changes in these microglia, you can actually tell, and you can see when the changes are occurring. And I will let Luke describe a little bit more of his transcriptional changes that he observed.
Yeah, so we saw changes in the inflammatory profile of these microglia shortly following benzene exposure, specifically in the NF-kappa B pathway. And then we saw changes in inflammatory cytokines, NF-kappa B itself being a master regulator of inflammation. And so the fact that it's changing in these brain immune cells shows that you get inflammation from benzene pretty quickly, transcriptionally.
Luke, can you maybe just break down that pathway a little bit for us? Right. So the NF-kappa B, why is it so important? Why did it stand out to you? And how specifically does benzene exposure activate that pathway?
NF-kappa B is a master regulator of inflammation. It's normally held in an inactive state in the cytosol, and then upon exposure to any type of inflammatory milieu, its inhibitor is degraded, and then it moves to the nucleus where it activates the transcription of multiple inflammatory genes.
Tell us about the knockout, why you chose that specific knockout, right? Because I think that's an important part of your strategy.
Gotcha. So after acute exposure to benzene, we saw activation of these NF-kappa B pathways changing transcriptionally in the microglia. So we then wanted to ask the question, could we rescue these animals from this metabolic dysfunction by altering this NF-kappa B pathway in the microglia? And to do that, we knocked out a protein called IKK2 in these microglia. And so this protein is normally involved in the degradation of the inhibitor that holds NF-kappa B in the cytosol. And so without this protein, NF-kappa B is held in an inactive state no matter what happens. So basically, it doesn't matter if you have this benzene in these animals that's normally triggering this inflammation. Without this protein there, NF-kappa B can't activate this inflammatory gene transcription profile. So we used a tamoxifen-specific knockout model where we're inhibiting this protein, and that stops the inflammation specifically in the microglia. And we found that this rescued these mice from this metabolic dysfunction caused by benzene. As to how benzene itself is triggering the NF-kappa B pathway in the microglia, I'm not sure we looked at that. We know, like Mariana mentioned, that The hypothalamus is specifically sensitive to environmental pollutants because it has kind of a leaky blood-brain barrier. So the benzene's getting in there. Benzene is a small lipophilic molecule. It's easily able to cross over cell membranes. So it's probably just coming out of the blood after general inhalation.
So in terms of the, again, so what we saw previously was that within the microglia, there is activation of cytochrome P450, which is a critical component of these VOCs. And it seems that the activation of the cytochrome P450 does have effect activating NF-κB signaling. But of course, absolutely everything will activate these as sort of a major inflammatory mediator, right, of so many different things. So I'm sure there's lots of different, upstream regulators that might trigger that effect. We did not specifically go into which of these potential upstream targets might be those that are activating the pathway. I'm sure it has to do more specifically within the metabolites of these VOCs in our cells. So yeah, there will be, it might be secondary, it is very much possible it's not primarily, but actually secondary. And this is how you already get this activation of NF-κB signaling. So we do not know that. And it might go both ways, right? It's absolutely possible because, it's difficult to tell. It's such a major regulator of absolutely any type of stressor. But I think that the critical component here is first that you can alleviate VOC-induced metabolic dysregulation, right, through the NF-κB signaling.
Right.
Second, it's not really trivial. You can say, okay, once you block NF-κB signaling in microglia, there are many other models, including, let's say, Alzheimer's disease models with obesity, models with such and such. And it's usually, can alleviate or even block the phenotype that you observe. And so you can say, oh, maybe that. But reality is that this is whole body inhalation, right? This is whole body exposure. These toxins that we're exposing in a relatively low concentration are capable of inducing and activating lots of different pathways. Inflammatory, inflammasome, for example, as just additional example, which will be a different downstream, activating pathway. Inflammatory, yes, but many different ones, right? So the fact that our knockout or inducible model in which we specifically deleted NF-κB in microglia later in life in these animals just before the exposure, and that was enough to alleviate the phenotype, that indicates, well, I want to say the critical role of this specific signaling within these microglia, also sort of including these toxic exposures, these environmental exposures, in this sort of cluster of other diseases, of other toxins, of other stressors that we already know are, functioning through NF-κB signaling. So now you have that and you're like, okay, so it doesn't matter that many other pathways probably were activated. It doesn't matter that lots of, other negative side effects were occurring from the inflammation perspective. This one and blocking this one was absolutely enough. And when you're talking about potential mitigation / pharmacological possible intervention, that already tells you that even if you have a question, oh, what else was activated? What else was going on? I'm sure a lot in the brain and outside of the brain, no doubt. But if that was enough, that's already, good indication for you that we can move forward, from that point on.
Well, that's an excellent point to end the interview. I'd like to thank Dr. Sadagurski and Stilgenbauer. Sorry, tongue tied on this Monday, for joining us today. It was, I should add a little disclaimer that Mariana and I have some previous collaborative work, but it was a real pleasure to be able to see you and see what's going on in your lab these days. I encourage the listeners to read the full article in the December issue of Diabetes. Thanks to you both for joining us.
Thank you so much again for the invitation. We had fun.
Yeah, thank you for having us
again. Bye.
Anytime. For today's interview, we're going to mix things up a little bit. Darleen couldn't make it, so Kevin's going to jump in, and we're going to discuss December's paper of the month titled Effect of Weight Loss on Skeletal Muscle Bioactive Lipids in People with Obesity and Type 2 Diabetes. We're joined by lead author Max Petersen, who is an instructor at the Division of Endocrinology, Metabolism, and Lipid Research at Washington University School of Medicine. Welcome, Dr. Petersen. Congratulations on your paper, and thank you for being a part of this month's podcast.
Thanks for having me.
So we always like to start with a little bit of an icebreaker. I'm hoping you can tell me your favorite fact about science.
So I have a 6-year-old boy now who's obsessed with dinosaurs, and I love thinking about just the sheer enormity of time that dinosaurs walked the earth. I think we see Jurassic Park and we think about dinosaurs as just this era on the earth when dinosaurs walked the earth. But there were 3 whole geologic periods, Triassic, Jurassic, Cretaceous. There's hundreds of millions of years that dinosaurs walked the earth. And it just puts, our small place in the world into perspective whenever I think of that.
Yeah, that's a good one. That's a good one.
Yeah, definitely. Hey, Max, we like to give our guests a chance to recognize any key collaborators or contributors to the work that's being presented. I thought maybe you could do that, as well as maybe you could give a little overview of the work that Dr. Klein is doing in his group that you're a part of, and how this project fits into the broader scale.
Thanks. Yeah, so this work is really a collaboration between the groups of my current mentor, Sam Klein at WashU, and my graduate mentor, Jerry Schulman at Yale. And the Schulman lab has expertise in lipid-induced insulin resistance in skeletal muscle and liver. Sam Klein's group has expertise in mechanisms linking obesity to metabolic disease in people. And, really the central question that a lot of the work in the Klein group is trying to answer is, why is it that obesity causes metabolic problems? And why is it that weight loss is beneficial for things like insulin sensitivity and multi-organ system function? And these are questions that are so fundamental and that we take for granted in clinical endocrinology, but are quite complex to answer at a basic level. One of the reasons that I think people think and has been investigated for decades that overnutrition or weight gain causes insulin resistance in the skeletal muscle is accumulation of toxic bioactive signaling lipids in the skeletal muscle and inside the myocyte. And so we had a unique opportunity here to study people who gained weight, developed type 2 diabetes, but then lost a significant amount of weight through interventions that we provided as a study team. We collected skeletal muscle biopsy samples before and after weight loss, and were able to assess those signaling muscle lipids to see if they changed in association with the dramatic improvement in muscle insulin sensitivity that the weight loss provided.
Great. So Max, let's get a little bit deeper into the paper here. This work tackles, as you mentioned, obesity-associated insulin resistance and the role of bioactive lipids. Can you give us some background on diacylglycerides and ceramides as they pertain to insulin sensitivity and glucose homeostasis, especially in the skeletal muscle there?
Sure. So it's been known for decades that accumulation of lipids inside the skeletal muscle is associated with insulin resistance in the skeletal muscle, but the triglycerides that comprise most of that lipid are generally not thought to be active in terms of cell signaling. They're a source of fuel, but they're not typically thought to directly cause insulin resistance. But there are certain lipid species inside the cell that can interact with various signaling pathways and potentially cause insulin resistance by impairing the process of insulin-stimulated GLUT4 translocation, which is ultimately responsible for insulin-stimulated glucose uptake. 2 of those signaling lipids are diacylglycerols, which act through activation of protein kinase C isoforms, and ceramides, which act through probably multiple mechanisms depending on their site of action within the cell. Diacylglycerols are interesting because they have 3 different isomers. Depending on which 3 of the carbons comprising the glycerol backbone have a fatty acyl chain attached to them, you can have either fatty acyl chains at the 1 and 2 position, the 1 and 3 position, or the 2 and 3 position. And these isomers have relevance for the signaling pathways that are downstream. Specifically, only the 1,2 isomer of diacylglycerol can activate protein kinase Cs and signal through that pathway. So we measured those different isoforms of diacylglycerol in this study in collaboration with the Schulman lab.
Very cool. Very cool.
Yeah, I thought it was quite interesting that you were reporting on those two different mechanisms with DAGs and ceramides in your study. And I think it was very interesting that the data appeared to strongly point to one of these mechanisms. Could you elaborate more on that primary observation and what it reveals about the role of these lipids?
Yes, so these people, we studied 14 people who lost greater than 15% of their body weight through either Roux-en-Y gastric bypass surgery or a low-calorie diet intervention. And so their BMIs went from 43 to 35. They all had type 2 diabetes, and their hemoglobin A1Cs went from 7.7 uncontrolled to 5.9 very well controlled. Their skeletal muscle insulin sensitivity measured by the gold standard clamp method doubled. So we know we improved muscle insulin sensitivity in these people. And the muscle bioactive lipids were as follows. So the diacylglycerols, all three isomers did not change in content after weight loss, which was contrary to our initial hypothesis, which was if these diacylglycerols are toxic lipid species that are inducing insulin resistance, we would predict that they would decrease after a weight loss intervention that improves insulin sensitivity. We did not see that. We saw no difference in diacylglycerols of any isomer, including the bioactive 1,2 isomer. We also saw no difference in total ceramide contents in the skeletal muscle, but this masked differences that we saw in individual ceramide species. Specifically, we saw reciprocal changes in the C18 ceramides, which are actually the most abundant muscle ceramide species, which decreased in key compartments after weight loss, whereas we saw reciprocal increases in a very long chain ceramide species, C24-1, such that when we summed all the ceramide species together, we saw no effect of weight loss. But again, this masked differences in individual species, which could have physiologic relevance.
Right. And if I remember right, you saw differences in subcellular locations of those specific species. Is that correct?
Yes, so we fractionated our skeletal muscle biopsy samples using a sequential ultracentrifugation method that was developed in the Schulman lab several years ago that allows us to isolate sarcolemmal fractions, mitochondrial / endoplasmic reticulum fractions. We're unable to adequately separate those two organelles, so we pool them as one, a lipid droplet fraction, as well as a cytosolic fraction. And specifically, we saw decreases in that C18 ceramide species, most prominently in the mitochondrial and ER fraction.
I think your study focused in on specific weight loss strategies. So I'm curious, with the emergence of these really highly effective incretin-based weight loss therapies, would you anticipate that these drugs might produce similar changes in skeletal muscle that were observed in the weight loss strategies that were used in your study?
So I'm not aware of any studies that have directly addressed skeletal muscle bioactive lipids, meaning diacylglycerols and ceramides, in people who have lost weight with GLP-1 receptor agonists. But we did study people who lost weight by two very different methods in this paper. We studied people who lost weight via gastric bypass and people who lost weight via low-calorie diet. And we saw no effect of the mode of weight loss on our results. They were broadly consistent across types of weight loss. So I would hypothesize that weight loss by GLP-1 receptor agonists would achieve a similar effect.
So, Max, I'm hoping maybe you can give us the 30,000-foot overview now, right? What insights are we pulling away specifically from your work regarding the diacylglycerides versus the ceramides and what weight loss does for those bioactive lipids in the skeletal muscle?
I think that these results actually dovetail with work we published earlier this year, which was getting at the question of why some people with obesity remain relatively metabolically healthy, whereas others are metabolically unhealthy. And in that study, which briefly we studied 20 people who had metabolically healthy and unhealthy obesity, perfectly matched for body mass index, age, sex, and measured various cardiometabolic outcomes to try to discern what distinguishes people with healthy and unhealthy obesity. And in that study, the mitochondrial ceramide content also came up as a very powerful predictor in the skeletal muscle of who had metabolically healthy obesity and who had metabolically unhealthy obesity. So to me, the 30,000-foot view here is these sets of studies really suggest that the mitochondrial ceramide content could be an important cause of muscle insulin resistance, and reductions in mitochondrial ceramide content could be part of why weight loss improves muscle insulin sensitivity.
I guess as a follow-up, if it's the mitochondria in the skeletal muscle, are there reflective circulating cells that you could use as a biomarker to say who might be obese and metabolically healthy versus someone who really needs to lose that weight?
Yeah, so we have performed some plasma lipidomics in our people with metabolically healthy and unhealthy obesity to understand if circulating ceramides, and specifically these C18 ceramides, which again are the most abundant ceramide species in the skeletal muscle, could be a biomarker of muscle C18 ceramide content. And there is a statistically significant correlation, but probably not a robust enough correlation to serve as a biomarker, a readout of what's happening inside the skeletal muscle. There has been some interesting work, not from our group, which has attempted to discover the mechanism by which mitochondrial ceramides might cause insulin resistance. And there have been in vitro and cell-based studies that have shown that mitochondrial ceramides can impair ADP-stimulated respiration, so mitochondrial function, as well as deplete coenzyme Q in cultured skeletal muscle cells. So I think additional work needs to be done to link those effects to insulin signaling per se and glucose transport per se. But I think our data provide some potential human relevance to stimulate future mechanistic work in that direction.
So Max, I guess one quick question. You shared with us that you finished your training and you're heading out on your own, right? So are you going to stay in the bioactive lipid field, or any hints for what you want to do as your own PI?
I've taken a real interest lately in nutrition research and the effect of different specific diets on metabolic function, both in people with obesity and also with lean adults. I think that stable isotope tracers, assessments of metabolic flux, provide a really powerful tool to follow metabolic function in people. And I want to use my clinical and scientific training to do mechanism-oriented studies in human subjects, but primarily probably using isotopes to trace glucose and lipid metabolic fluxes.
Very cool. Well, Max, we want to thank you for joining us today, for sharing your expertise, and for discussing your recent work on the role of skeletal muscle biolipid changes associated with improved insulin sensitivity after weight loss. I encourage our listeners to read the full article in the December issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams.
I'm Darleen Sandoval.
Hey, and I'm Kirk Habegger.
Today, we're joined by Dr. Kathleen Page and Dr. Jasmin Alves. Dr. Page is an associate professor of medicine at USC's Keck School of Medicine, where she co-leads the Division of Endocrinology and Diabetes and directs the Diabetes and Obesity Research Institute. Her research focuses on the causes of obesity and diabetes, especially neurodevelopmental pathways, to improve prevention and treatment. Dr. Alves is a postdoctoral fellow with Dr. Page and studies how early postnatal factors are related to metabolic outcomes in youth exposed to gestational diabetes. They're here to discuss an article in the December issue of Diabetes, discussing work funded by the American Diabetes Association Pathway to Stop Diabetes Initiative that Dr. Page received in 2014. Welcome, Dr. Page and Dr. Alves, and thank you for joining us.
Thanks for having us.
Before we get into the science, we always like to start with a little bit of an icebreaker. If you were not focused on obesity and diabetes research, is there another passion or career you could see yourself pursuing, maybe something completely outside of science? We'll start with Dr. Alves and then maybe move on to Dr. Page.
I do feel like I have my dream job right now, getting to research diabetes. I think if there was something alternative, I know this is random, but I think I might enjoy being a police officer.
Oh, wow.
All right.
I've always thought, oh, wow, that seems like a fun job, getting to be out and about in the city and helping people.
Yeah. Any family link?
Surprisingly, no, I just love police dramas on TV.
Yeah, I was going to say, Jasmin, with your postdoc mentor on the line, that is the correct answer, right? You already have your dream job.
Yeah, well done, Jasmin. No, it's funny because I've always loved science, so of course I would probably choose something science related. I started out in exercise science and have a graduate degree in physiology, but maybe it's all of my watching law shows, but I think if I didn't do science, I would have gone into law. So I would have been a lawyer, you would have been
Wow.
a police officer. We'd still be a good team.
I'd watch that sitcom. Dr. Page, this work was funded by the American Diabetes Association, the Pathways to Stop Diabetes. I was wondering if you could comment on the importance of this initiative and what it's meant for your research goals in your career.
Yeah, I mean, the 88 Pathway Award was given to me in 2014. So I was part of the initial cohort of pathway investigators. And I can tell you that this was a time where I think there was a lot of skepticism in the field about neuroimaging, and people didn't necessarily think that we were going to be able to do neuroimaging in children. But I think, they invested in us, they believed in us, and I think it's really paid off because the field has really expanded since we initially started this cohort. And I think we've shown really important findings. So I think without the 88 Pathway Award and taking a risk, supporting me during an earlier stage of my career, I wouldn't have been able to initiate this cohort. And that has really led to instrumental findings and also longitudinal studies that I think will be really important going forward.
Yeah, I must say I'm excited to see this work and the cohort that's being studied continued on, whereas a lot of times, you think about research projects to where they will terminate once funding is stopped. And so it's nice to see that this work will be continuing on and even more developments might be coming out of it related to diabetes research. So it's very exciting.
All right, awesome. Let's start talking about science. So, I'd love to hear more about what drove you to this particular field, in particular maternal and fetal health. So, Katie, we'll start with you.
Yeah, I've always been interested in physiology, and I think I started out in a lab where we were doing a lot of work in diabetes and in understanding how the brain regulates glucose homeostasis. But I started reading a lot of articles about early life programming and developmental programming. And so then I started to think, how could we sort of understand these brain pathways at an earlier stage of life? And so that's how I sort of started getting interested in the field of maternal fetal programming and just really reading mostly preclinical work, studies in animal models that had shown that animals who were exposed to diabetes in utero had changes in the development of the hypothalamus that in turn led these animals to develop obesity and diabetes later. So that was really what sparked my interest in this field.
Awesome. And Jasmine, besides the fact that you were drawn to work with Dr. Page, what drew you to this field?
I've always been really interested in the field of obstetrics. My great grandmother was a midwife, and it's a passion that's been passed down through my family. The idea of women being this magnificent creature that gets to bring life into the world and form the health of that child for the rest of their life. So that's really where my passion started for the subject.
Awesome.
So, Katie, you described the use of the brainchild cohort in your article, and you really emphasize how some of the regions of the brain that are involved in appetite and reward control are affected by that intrauterine exposure. Can you tell us a little bit more about the brainchild cohort specifically, and then some of those key changes that you observed?
Definitely. So we started this brainchild cohort almost a decade ago. So the 88 Pathway Award was really pivotal to developing this cohort. So right now we have about 250 children. All of these children were born at Kaiser Permanente, Southern California, which means that we have very detailed information about the children and their mothers during the pregnancy. So we know if the mom had gestational diabetes or normal glucose levels. We know the glucose levels that were used to diagnose gestational diabetes. We know the mom's height and weight throughout pregnancy. We know a lot of information about the child and the mother during that pregnancy. So that's the cohort. We have 250 children. Half were born to mothers with gestational diabetes and half were not. And we've done very detailed metabolic profiling on those children at the age, starting at age 8, and then now longitudinally, we've been following them. We also do extensive neuroimaging in these children, so we can really address the question of whether exposure to gestational diabetes in utero leads to changes in the development of pathways that in turn lead to higher risk for metabolic diseases. And so some of the key findings that we've had so far in this cohort, the initial abnormality that we have seen in these children at age 8, prior to any differences in body weight or body composition or glucose homeostasis, is a change in the development of hypothalamic pathways, the way that the hypothalamus is responding to glucose, and also a marker of hypothalamic gliosis or injury to the hypothalamus. That's the first initial finding or difference that we see in the children who were exposed to gestational diabetes during early fetal development. And those are linked to increases in weight gain over the following year. So we are now following these children longitudinally so we can identify whether that initial finding is going to be related to changes or susceptibility to the development of obesity and type 2 diabetes, as was seen in the animal models.
So I guess as a follow-up, I have a question about the behaviors, right? So are there specific behaviors or food preferences or eating patterns that may align with those brain changes that you observed?
Yeah, I think that's a really important question. We've done a lot of work to really identify dietary preferences and dietary intake in these children. So we follow what we call 24-hour recalls every single year in these children. And we're looking to identify whether there are differences in their eating patterns. So far, what we've seen is that the children who were exposed to gestational diabetes, particularly during early fetal development, had greater energy intake even prior to the development of obesity early on. So we don't know yet. We haven't followed them longitudinally. We haven't analyzed the longitudinal data to look at dietary preferences, but I think it'll be really interesting to see whether they're consuming more sweet foods, what their dietary preferences are. And it may be that the women are empowered with the knowledge that these children are at greater risk. And so they may actually be changing their eating behaviors earlier in life. So what we'll be looking to see in the next decade, what's happening with these children and their eating behaviors.
Yeah, very cool.
I think one fascinating part of the study is how timing and the severity of the maternal diabetes exposure influences the child's risk for obesity and type 2 diabetes. I'm curious if you could go into a little bit more about what you found about what's so critical about the timing and that severity of exposure and how it affects the different aspects of the brain and metabolic development.
Yeah, definitely a really important part of our findings. So what we found was that gestational diabetes as a whole, so if we look at all comers, all of the children who were exposed, we didn't find a significant impact on brain development and risk for disease. It was only the children who were born to mothers who had gestational diabetes diagnosed less than 26 weeks, so really in the first trimester. Those were the children that were at risk for changes in brain development and obesity later. So what we think is happening is that during that first trimester, that's when the brain is really rapidly developing. So the timing is probably important, but also the severity. So the women who had gestational diabetes diagnosed early in pregnancy had higher glucose levels than the women who were diagnosed later. So we know that because we have their glucose tolerance tests. And when we adjusted for the severity of glucose, it attenuated our results, suggesting that the degree or the severity of hyperglycemia may be playing a role in the susceptibility for disease.
Very good.
I'll start with Jasmin on this next question, since you're the future of the field. With gestational diabetes on the rise, as with all cases of diabetes and obesity, what are the implications for public health? And do you think of or dream of any potential ways that you could guide strategies to prevent or reduce diabetes risk?
I'm so glad you asked that question. That is actually my long-term career goal, is actually to translate some of the mechanistic findings from Katie's work into practical interventions. We've had some really interesting studies showing that physical activity is associated with better health outcomes in the children exposed to gestational diabetes compared to children who are not physically active that are exposed, suggesting that physical activity could be one potential avenue for an intervention. So that is definitely something I want to pursue, given that there are these rising rates of obesity and type 2 diabetes in children.
Yeah. How do you convey the messaging, right? Because if mom has this health, right, this maternal health problem, you don't want to blame mom for the issue. And so how do you work the messaging here? And you can both answer this question.
Yeah, I think it's about empowering people with information, right? So we're trying to identify what are potential risk factors and who are the children and the mothers who are at highest risk, and what can we do to help those women and their children shape better health trajectories at an earlier stage of life. So by identifying and understanding the causes of these links between gestational diabetes and higher risk for disease, we can both give the knowledge to the women and their children so that they can implement evidence-based practices that will help them set the stage for better health in themselves and their children. But we may also influence public policies so that we can give them the resources and the support that they need to, help their own health and to help the health of their children, which will hopefully set the stage for better health across generations.
Yeah, great. So, many of our listeners are researchers. And from a research perspective, can you maybe tell us about some of the challenges that you faced in studying the genetic and prenatal influences on obesity and diabetes? And maybe touch on the population that you chose to study, right? There's a fair amount of diversity, if I remember right, in your article.
Yeah, so I think that metabolic diseases, of course, are influenced by a number of factors, including genes and environment. So trying to disentangle that is challenging. But I think there are two lines of evidence to support an independent effect of the intrauterine environment. And those two lines of evidence are one, sibling studies. So siblings who are discordant for exposure to gestational diabetes, meaning one sibling was exposed to the mom who had diabetes during pregnancy and the other sibling was not. The sibling who was exposed has a significantly higher risk of developing diabetes later in life than the unexposed sibling, suggesting that even though they have shared genes and shared family lifestyles, there's still this independent effect. The second line of evidence is evidence in families. So the mother who has gestational diabetes, the child has a higher risk of diabetes than if the father had diabetes during that same time period. Again, suggesting some independent role of that intrauterine environment. So I think there are studies to suggest an independent role of the intrauterine environment, but certainly genetic factors are important. Prenatal factors are important and postnatal factors are important. So understanding how we can set the stage for better health in these children at an earlier stage of life, independent of their genes, is going to help hopefully improve the health trajectories of women and their children.
I think it's also exciting that I believe that the study that you were currently focused on was in the 7-8 year range, and you're continuing on this study to look at these children as they age. Are there specific outcomes, long-term outcomes that you're particularly interested in tracking in these children?
Yeah, so in the perspective paper that will be coming out in December, we have a conceptual model. And this is the conceptual model is that these children who are exposed to gestational diabetes, particularly during early fetal development, may have changes in brain pathways, particularly in the hypothalamus, which may increase their susceptibility for obesity and diabetes. But they haven't yet become obese. So what we're doing now in our longitudinal studies is following the evolution of metabolic diseases at a high risk cohort. So we can understand what are the factors that may accelerate these risks and what are the factors that may actually ameliorate those risks. And so by identifying those factors, we hope to implement them, particularly in high risk children, and potentially give us information that may help us set the stage for women to get better health prior to pregnancy and early in pregnancy so that we can sort of stop this vicious cycle of diabetes in women and children.
Nice. And as we wrap up, one of the main takeaways I think from your work is the impact of maternal health on future generations. If you could share a message for healthcare providers, researchers, maybe even parents about the importance of maternal health in preventing obesity and diabetes, what would you say?
I think I might echo Katie's sentiment earlier that knowledge is power. So I think just simply informing families of these intergenerational risk for obesity and type 2 diabetes is how they can make a difference.
Nice. Thank you, Dr. Page and Dr. Alves, for joining us today. And thanks to our listeners for tuning in to this episode of Sweet Talk. Be sure to check out the article titled Neurodevelopmental Pathways to Obesity and Type 2 Diabetes, Insights from Prenatal Exposure to Maternal Obesity and Gestational Diabetes, a report on research supported by Pathways to Stop Diabetes, available in the December 2024 edition of the journal. And as always, please like, follow, and subscribe for more DiabetesBio content.
Thanks to all of you for joining us on the DiabetesBio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.